Great, thank you. So thank you all so much for joining us tonight for this important webinar, where we'll be exploring the science of the algal bloom affecting South Australian coastlines. My name is Alfie Lem and I'm the coordinator at the Port Environment Centre, and I will be moderating tonight's discussion. First of all, I'd like to acknowledge that I'm coming to you from Kaurna land, and I'd also like to acknowledge the lands of the Boandig, Ngarrindjeri, Narungga, Nukunu, Bangala and Nao people, all of whose lands and waters are affected by this algal bloom. I'd like to pay my respects to their elders past, present and future, and acknowledge their ongoing connection to their lands and waters. I want to acknowledge the work of First Nations people in this space, including the Walk for Water event that took place last Saturday, where people walked from Victor Harbour to Goolwa to honour their sea kin that are affected by this bloom. So this webinar is brought to you by the Port Environment Centre. We are a community-led space in Port Adelaide and we operate as an independent not-for-profit. We are one of 9 community environment centres in the Greater Adelaide region, and our focus is on coastal and marine environments. We receive our funding from Green Adelaide, the City of Port Adelaide Enfield, and through the Suzanne Elliott Charitable Trust. We support local environment groups and run events to help people learn about their natural environment and build skills to live more sustainably. You can come visit us. We're at 27 North Parade, Port Adelaide, and we're open Monday to Friday, 10 till 3 p.m. And you can come visit us, and we've got a bunch of free resources. You can ask questions, or we can help you with your environment project or idea. You can find us on Facebook, Instagram, or via our website. So yeah, just get in touch with us anytime if you have questions. And tonight, as you all know, we're here to talk about the algal bloom. We've seen algal blooms before on our coastlines, but the duration and extent of the current bloom is really unprecedented. The algal bloom is a statewide issue impacting communities at all levels, and not only is it affecting people's ability to get out onto the beach and into the water, but it's affecting industry and research too, and anyone that cares about our marine environments has been affected. We've seen a lot of heartache in the community from witnessing the die-off of our precious marine life, so we wanted to give this opportunity for community to come together and hear from experts who work in this area every day, and to answer questions that have largely gone unanswered, and to reach out to people from across the whole of South Australia. So we've got an incredible panel tonight with 6 speakers, and they're here to speak to the science of the algal bloom and how you and other community members can get involved in monitoring and recording the event for citizen science. And our first speaker tonight is Janine Baker. Janine has worked in marine research for 35 years with roles in government, NGOs and universities, working on projects focused on monitoring and conservation of a variety of marine species throughout South Australian waters. Marine Janine is currently managing citizen science projects focused on introduced species, sea dragons and nudibranchs, and delivers workshops across the state to help involve local communities in marine monitoring. So I'll hand over to Janine and she'll give us an introduction to the science behind the algal bloom. Thanks so much, Janine. You just unmute yourself, Janine.
Is that better? Hello. Yep. Thank you very much, Alfie, and thank you to all the speakers here tonight, and thank you to everyone for tuning in. I'm going to share now and hope there are no technical hitches. Let's see how we go. All right, can you see that?
Yep, looks good.
Okay, that's fine. So thank you very much, everybody. I just wanted to have an introduction here, and these lobby messages keep coming up, which I really hope they would. We're going to have a little chat about some of the precursor conditions, which Jochen will discuss in a lot more detail later, and some of the observations that people have made in different areas around the state. So this algal bloom really came to attention in March when people on the Encounter Bay Surf Coast started getting sick from the aerosol and the foam from the algal bloom. But it's quite possible that this has been going intermittently for a fair bit longer, because there's evidence from around the state of fish kills over summer, but also last year. I'll give an example here of the southeast. So during annual period in summer of the Bonnie upwelling, when deeper nutrient-rich water comes to the shallows, it was very cold in the coastal waters there, about 11 degrees in the southeast, and chlorophyll maps show that there was quite a large concentration of chlorophyll, which is normal, and this drives productivity. However, fishers at the time in the southeast were noticing fish kills in a number of areas. So some of the things that they were noticing, such as masses of octopus washing up on beaches, masses of dead sharks washing up on beaches, and a lot else, really mirror what we're seeing now in 2025. So the first of those was in January 24 down near the 8 Mile Creek area, and that also coincided with a time of nutrient-rich runoff from 8 Mile Creek after heavy rain. So I think at the time it wasn't considered that there may be a possibility of something going on in the marine environment as well as that nutrient-rich runoff. But one of the fishers down there sent these photos of some spotted stingeries that show the same red blush that thousands of animals around South Australia that are washing up is showing. And there was also mass death of cockles in that year as well. So fast forward to January 2025, more deaths started being seen, and some really quite unusual things, like some sharks, whales washing up. Here's an example of one of the beaked whales at American River. And even mortalities from Encounter Bay, where the mass bloom-related mortalities started to really become obvious in March. They were starting to be recorded right back in February, and possibly also in Northern Kangaroo Island as well. So we're trying to contact community in that area to see if they do have some retrospective records there. So as I mentioned, March was really the key time when all of this hit the media. At the time, there were mass mortalities in the southeast, but also in particularly in the coastal area outside of Encounter Bay. And I just wanted to put some of the chlorophyll maps up here from the time, but also with a caution, because this is false colour satellite imagery. So the areas of high chlorophyll concentration are the reddest areas, but interpretation must be cautious, particularly in shallow areas, because sediments in bays, for example, or what is known as CDOM, coloured dissolved organic material, or even bottom reflectants in really shallow bays, they can show up bright red as well, even in areas where there are no algal concentrations that are of concern. But of interest here is these three periods in March and April and May, with the circled areas. They actually mirror what people were seeing in terms of many, many hundreds of animals of all trophic levels washing up. So we might go through that in just a little bit of detail here. So from the surf coast of Encounter Bay, so particularly outside of Encounter Bay and all the way through Tunkalilla towards Cape Jarvis, and also the Murray Mouth area, many of you would have heard of the mass death of the Goolwa cockles as well during the earlier part of autumn. So what people were noticing first along that Encounter coast, apart from people who are on the beach or in the water getting sick, there are a lot of dead octopus washing up on beaches, plus a whole lot else, a lot of different fishes. And Brad will, from OzFish, will go through some of the data summaries later about that. And also really unusual things such as seals washing up with bright red all over their bodies. And that was, I guess, a sign of things to come. And at the time, it was possibly also in the Kangaroo Island area, in the northeast area in March, but by April, the bloom had apparently spread in a northwesterly direction, and there was a lot of work done by SARDI and other people in government to track that, and a lot of hydrographic modelling was done, and also following the progression of the bloom according to currents and wind patterns. So Investigator Strait is this area here. Some of you who live in that area or who work in that area will be very familiar with the deeper waters in Investigator Strait. But on both sides of Investigator Strait, Southern York Peninsula and Northeast Kangaroo Island, there was a lot of mortality in April. There also was on the southern part of Kangaroo Island, but unfortunately it's very hard to get any kind of visual information from southern KI. Fishers were reporting things such as hundreds of dead cuttlefish and fishers observed when they are fishing offshore, but fishers are very busy people when they're out on the water, so they often don't have cameras with them or phones, etc., to take photos of this. So a lot of that is anecdotal information, and we're asking people around Kangaroo Island if they have information from any of the months from summer to the present to provide information from anywhere on the island. It was particularly difficult time for York Peninsula as well, with thousands of rays and fish and many other things washing up. So the progression of the bloom showed. I also wanted to mention here in April, it hit Edithburgh, and Paul McDonald, a cinematographer and photographer, will be talking about some of the impacts in the Edithburgh area in April, because Paul's footage and from a couple of other divers in the Edithburgh area is some of the very limited information we have so far about what has happened underwater. And we know that there are significant underwater impacts. A lot of it is just not being recorded. And a lot of what we know in terms of mortalities is from what is washing up on beaches, which Brad will discuss the projects involved with that a little bit later. I'm nearly finished now, but I did want to mention, because these examples here are right up until the present day, so up until the 28th of May, there was a timeline progression of things washing up. So in the western side of Gulf St Vincent, so eastern side of Yorks, death started to be seen around the Edithburgh area, and progressively over the weeks further and further up the coast, up to at least Ardrossan now. So some of these photos here are things that have been washing up. So why is this happening? I'm not going to discuss in too much detail, but we know that a couple of toxic Carenia species, which are small dinoflagellates, are here, and also a related one called Carlidinium. They've been blooming since at least summer and certainly over autumn. So globally, the examples of these blooms are showing that they can last for months, unfortunately, and cover thousands of kilometres. So some of these blooms have happened in other countries. They have been devastating. This is particularly long-lived, these kind of blooms, and they can actually travel up and down the water column. So they're not just shallow blooms. They can move over tens of metres. Here's an example of one of the species. This is Carenia mikimotoi, and the other one, Carenia papillonacea, comes from the French papillon for butterfly, because the cells look a little bit like a butterfly shape. The full suite of species hasn't yet been publicly available. I'm not going to talk too much about the conditions, other than to say that over autumn we had incredible temperature anomalies, 10 degrees above average in the air, 3 degrees above in the water, huge nutrient sources from the upwellings and possibly from other sources. Upwelling is known to be a precursor in other countries.
Sorry, Janine, we're just
yes.
running out of time on your section here. If you just
Sure.
want to wrap it up.
Okay, will do. Okay, so just very briefly about Karenia, and Ian will talk about this in more detail later. So Karenia can kill in many different ways. Some of them are there, both the whole cells and immune reactions and destruction of gills, harmful impacts and organ damage. Here's an example of Karenia. Normal gill tissues, and this is the gill tissue that's been exposed to Karenia, which actually breaks down the gill. So unfortunately, these algal cells, when they're blooming, they can reduce oxygen levels during the death process when bacteria builds up, and seafloor species can die from suffocation and also exposure to the toxins, as well as animals in the water. And unfortunately, the cysts can be stored in sediments as well. I'm not going to go too much onto the impacts because Ian is going to talk to this about the damage, for example, that is being seen in some of these animals in terms of damage to blood vessels. There is bioaccumulation issue. This is particularly serious because it means there will be long-term impacts here in the South Australian marine environment. And thank you very much. I'm going to have a chat a bit later about some of the specific species. So thank you very much, Alfie, and
Thanks.
Everybody. I will stop
Thanks
sharing
so much,
now.
Janine. Yep. Yeah, just stop sharing your window. Great. Yeah, thank you so much. It's really interesting to learn about how the bloom progresses over time, and it really highlights how important it is for monitoring so that we can see where the bloom's starting and where it's moving to, and just know how it's behaving in our waters. So next up, we'll hear from Paul McDonald. Paul is a professional underwater photographer and cinematographer who started diving in Adelaide in 1990. Paul is passionate about diving and has many beloved spots in South Australia and further afield. Paul is a certified PADI digital underwater photography instructor and dive master, and he is the proprietor of Down Under Pics and Edithburgh Diving. Paul will share some footage with us taken at Edithburgh Jetty and discuss the subtidal impacts in that area since April. Thanks so much, Paul.
Thanks, Alfie, and thanks everyone for joining us tonight. I'll just share my screen. Hopefully everyone can see that first screen.
Looks good. Thanks.
Just in addition to Alfie's introduction, I've been diving for 35 years, over 1,750 dives and nearly 1,900 hours. And particularly at Edithburgh, which is one of my favourite spots, 312 dives and 420 hours. When the bloom hit Edithburgh, towards the end of March, everything was normal. We arrived back there on the 4th of April, on Friday, and we could see something wasn't right when I went down the jetty. I was going to dive with someone, but we didn't even bother to get our gear out. Perfect weather conditions, would have expected at least a minimum of 10 metres visibility, but you could see the viz was really bad. On the 5th of April, we stood on the jetty and by the steps, and we could look down and we actually saw cockle shells on the bottom the fishermen would have thrown in. And we thought, it can't be that bad. So we got the gear on. Elizabeth and I and some others got in the water. I put my head down and looked down, and the viz was less than a metre, and it was a horrible dirty brown colour. So we got straight back out. But some divers reported dead animals that did go and dive. On a Sunday, I dived with Elizabeth. We did the dive after some discussion with the Yorke Peninsula Country Times, and to get some photographs for them for the report they wanted to do on the algae bloom. Again, it was still perfect weather conditions. We would have expected 12 plus metres viz, but the viz was about a metre and still a dirty brown colour. There were dead animals everywhere. And with a friend of mine, David Place, we've been monitoring a rodless anglerfish for over 3.5 years. It's been in the one spot, hasn't moved more than a foot in that 3.5 years. We'd seen it breed 3 seasons, and we couldn't find that, and we were fairly heartbroken. But just some photos from that dive. As we dropped down, on the pile, on the jetty, on the steps. As we were getting towards the bottom, we saw this dead blue ring octopus hanging off the weed, and it was, it was a foot off the bottom, and it was just wafting in the current, hanging on, but clearly very dead, but hadn't started decomposing yet. We went about 20 metres, and there were dead octopus, cuttlefish, squids, etc., all over the bottom. This was also near the steps. I think it's a lightning volute or some type of volute, laying there dead. Another blue ring, and again, we didn't go more than about 20 metres, Elizabeth and I, just to witness this horrible stuff. And this is just a selection of many photos. Lots of dead fish, dead sea cucumbers. So I said, lots of dead octopus of different species. That I believe is a sand octopus. And this one half buried in the sand. So that was our sort of first introduction, and we sent some photos off to the York Peninsula Country Times and also put them on our Facebook page, and ABC News Breakfast took an interest, and I did a interview with them that they played that ran for about 6 minutes. That was Country ABC, and also did an interview with SBS, and that was around dovetailing with the Gulwa cockles mortalities. Then I went back on Friday the 18th, which was Good Friday. I dived with David Place, a good friend of mine who also has a house over at Edithburgh. Again, perfect weather conditions. This time the viz was much better. It was around 2 metres. We would have expected 12 plus again. The water was a greeny brown colour. It wasn't as bad as it was the first time. I took some video under the jetty for the first hour of that dive, and then the second hour, Dave and I did a search for where we knew sea dragons were. They were out off the jetty in various locations, and there were 7 of them that David or I had known about. And then we did a second dive for photography, and so I've got some short video clips coming up and some of the photographs. Again, many dead and decomposing animals. Most of the jetty piles were bare wood, and in 35 years of diving Edithburgh, I have never seen the wood. All the soft corals and sponges were laying on the bottom and decomposing, or they were just hanging on. And then when we did the search for the sea dragons, which was unsuccessful, about a third of the corkweed cover was left. Normally it's quite prolific around Edithburgh, but what was left looked sick, and the rest was lying on the bottom, which was really horrible. But we did find several nudibranchs. We found some anglerfishes, and we found a Maori octopus, surprisingly, quite a large one, that was out walking around, and several other small fishes that were alive. No big fishes living. So here's the first video. I can run that. And so that's all the bottom structure that's dying, all that grey stuff. And on top of it was, if you touched it, it was like a white powder. And hopefully the scientists can talk about that some more. So again, and you got an idea of what the viz is like, but it was really, really horrid. Some dead octopus. Sorry, I think they're actually squids. And a lot of the particles that were in the water, I think, was that white powdery stuff as well that we were seeing. And here's one of David telling us all exactly what he thought of the place, and this was pretty much 2 weeks after it hit. And the timing of early April of that first dive certainly aligns with Janine's slides earlier about it hitting early April to the bottom of York Peninsula. So we didn't set this up. We came across these, I'm not sure whether they're squids or octopus or both, all together. in in state of bad decomposition. And a fish there, I assume it's a goatfish, but I'm not really sure because of the level of decomposition. And this is again 2 weeks after. Scallop there and a dead crab. And I think that on the left, I'm not sure what it is actually. Scientists might be able to help me. That's a codium. That was a rather large codium when it was alive, probably almost the size of a soccer ball or netball, but very, very dead. The other thing is sponges. They were skeletal, and if you actually managed to touch that or accidentally touched it, it just crumbled away. And there was lots of that. But of course, found some nudibranchs, and so there's one there. And we found some anglerfishes alive, which was really good. So that's all from me, thanks. So I'll stop sharing. And yeah, I hope that sets the scene of what it looked like under Edithburgh Jetty.
Yeah. Thanks so much, Paul, for sharing all of that. Those photos are heartbreaking, and it's really clear that you're deeply moved by the devastation caused by this bloom. And my heart goes out especially to that fish and that nudibranch that are there and stuck in this nuclear wasteland, essentially, without their whole community. And thanks for getting out there anyway, despite it being horrible, and documenting the death of these beautiful creatures and sharing them with us tonight. So after that very heartbreaking images, we're going to hear from Associate Professor Jochen Kaempf, who is a professional oceanographer with Flinders University. Jochen is an expert of the oceanography and flushing of South Australian gulfs, and his research led to the discovery of the Great South Australian Coastal Upwelling System, which hopefully he'll explain to us all tonight. He is a fellow of the Australian Meteorological Oceanographic Society and won the prestigious Jill Hudson Award for Environmental Protection by the Conservation Council of South Australia. Thanks so much for joining us tonight, Jochen.
I hope I can cheer you up a little bit with my presentation, because I also want to focus a little bit on the positive points. Okay, you can see a little outline of my presentation, and I will talk a little bit about upwelling, some facts about the Carenia Mikimotoi bloom, some oceanography, and I will also talk about the marine heat wave. What about the marine heat wave? What does it have to do with it? Starting with, there are good and bad algae blooms. Okay, so if you hear algae bloom, it's no reason to freak out immediately, because there are the really good ones, and it's shown here on the left-hand side. It's mainly diatoms, a diatom bloom in the Bonny upwelling, Bonny Coast upwelling, and you can see these structures, the light structures in the ocean. And why it is good is because it's actually the food source for zooplankton, then going into fish. It actually is the foundation of the entire marine food chain. Okay. On the right-hand side are the bad ones. The bad ones are the ones, that rather kill. They don't serve as a food source, but there are harmful algae blooms. And the Carenia Mikimotoi is one example shown at the top, but there are other ones. The red type is a very close cousin of the Carenia Mikimotoi, and that's also the blue-green algae blooms that we had in the Murray River. Okay, and you can see how closely linked the initial outbreak of the toxic algae bloom is actually, how close is that situated next to the upwelling. Okay, the good one and the bad one is actually very, very close together. And all these blooms are initiated by an excess of nutrients. So it's a nutrient, okay? Nutrient source. Okay, let's get on with the next one. So in satellite images, you can actually track the algae blooms. Okay, and here's one example of, it's just a time series from left to right, shows you the development of an algae bloom, of a true algae bloom along the Baltic coast. Okay, and you can see how it's slowly, the red are the it's a high concentration of a true phytoplankton bloom in the ocean. Okay, and as Janine mentioned, there's also fake blooms, and you have to be very careful with the interpretation in very shallow water. Okay, but I will not highlight that again. But now, what's the positive point of that? There's a number of these upwelling systems along the southern coast of Australia, and these upwelling of the production of phytoplankton, of algae, is actually the reason why we have such a productive and fascinating systems. And I show that's the reason why we have the tuna, the sharks, the whales, the dolphins. Okay, and that's why we are so passionate, and that's why the toxic algal bloom event is so sad, because we have that very productive upwelling system, and the upwelling happens every year. Okay, so this is to try to give you a little bit of a positive feeling. That's the only reason, not the only reason, but that's the reason why we're so devastated, is because we have these truly amazing range of animals. Okay, so now some facts about the Karinia mikimotoi blooms. And they're one of the most notorious ecological disasters with massive kills of valuable fish and shellfish in China, Japan, India, and many other regions. There are many subgroups. They're found worldwide in a wide temperature range between 13 and 25 degrees Celsius. Also they also have a wide tolerance range for salinity, from low salinities to seawater salinities, and they have optimal growth range within 17.5 to 22.5 degrees Celsius. But hang on. They actually can grow in relatively cold water. And this is an important information. If you find something like this, you know, it's not, this species does not grow better if it's hotter than 22.5 degrees Celsius. And that's already an important link to the marine heat waves that I will feature in a few slides. Okay. First described in 1935, and there's also references to blooms earlier than that. Okay, and I can highly recommend this paper, which is written here at the bottom. It has all the recorded events, and it's well known that the blooms can last more than 2 months, and these authors say they are always of extremely long durations. And why? Because there is no control of these blooms. Once they get started, there is no real control. They spread through currents and possibly long-distance bioinvasion and transport, and they're also recorded in upwelling regions. All right. Okay, so let's look at the average sea surface temperature maps. I want to talk a little bit about the oceanography, what happens during that time from March to April to May. There's a number of things happening in the South Australian regions, in which is illustrated here in terms of sea surface temperature. Warm is shown in red colors, is more than 20 degrees Celsius, and the blue is the cold water, less than 15 degrees. And if you look on the left side, you can identify the upwelling. Upwelling is a process lifting cold water, nutrient-rich cold water to the surface, and that's how you can see it. You can see that blue, we call it an upwelling plume. This is the Bonny upwelling plume. If you see something like this, you know, oh, it's nutrient-rich water, and it also spreads to the northwest. There's a little white arrow, so it actually cools the ambient regions. What you also have is you have warm hypersaline Gulf waters, the red areas. And what it is actually water that's, the Gulfs are actually isolated from the open ocean. That's why they are so, they can be very warm during that time. So in April and May, what happens in this region is there's warm water coming from the northwest, and it's an extension of the Lewin current. It actually brings nutrient-low warm water into the region. And this is now here the normal situation. It's averaged over 20 years, and it's not the same year. It varies from year to year, but before I talk about the variations, I want to focus on one more feature and a little bit more of a highlight in March. This is about the connectivity. So you learned that the algae bloom, where it has started, and just at the bottom of the Florida Peninsula, but there are now processes that can distribute the toxic algae bloom. And the starting point is that there is a very strong tidal currents in backstairs passage, shown by this black arrow, distributing, mixing water across backstairs passage. And there's also relatively slow distribution, which you can sort of see in the temperature shading that distributes it into Investigator Strait and upward on the western side of the Gulf St. Vincent. No connectivity to Spencer Gulf. This is what we can infer from looking at satellite images of sea surface temperature, which are much more accurate than chlorophyll pictures. So this is reliable information. And this one agrees with the spread. And as on a positive note, generally there's little exchange between the gulfs, and that's why the waters are hypersaline and warm. And there's only a little bit, and luckily the spreading is not so fast, because imagine that you had a fast spreading, then the bloom in Gulf St. Vincent much earlier and with much more devastating effects. All right. So what I want to do just at the end is I want to look at the year-to-year variations. So what I want to do is I just look at that little square area. I look at satellite data of the sea surface temperature, summer month, but now want to look at what happens from year to year in this area. And this is a good indicator. So it's not directly in the upwelling system, but it's still sort of through the tides, you get some signature there. And that will be very, very interesting. Let's have a look. So this is now a time series. On the left is 2003. That's when the good quality satellite data started to become available. And on the right side is 2025. And it shows 4 months in different colors. You see January in yellow, green is February, red is March, and the blue is April. And at the top, the title, you can see the average temperatures. For example, and just focus on the red March, the average temperature, if you average all years, is 21.6 degrees Celsius. And now the graph shows you whether it's colder than that in a particular year or warmer. And you can go and follow just the red bars, and you can see sometimes it's colder by a degree. Sometimes it's warmer by a degree. And let's just look at some examples. A marine heat wave reported in 2013, also causing fish death in different regions. And you can see the satellite map of the entire region. Okay? And then you go, but there's also areas where it's colder. So what happens during these times, like one example, last year was one example, but 2020 was also one example. You can see it's a negative bar. It goes down by 1 degree. And these are the times when you have a stronger upwelling, less warm water coming from the northwest, stronger upwelling. And in 2025, again, a marine heat wave. We just had another one, the second one recorded. and look at the different colors. Your yellow bar is pretty much that. You flip back and forth between 2025 and 2013. Yellow bar, same length in January, February, sort of the same. In March was warmer in 2013 by half a degree, but this one in 2025 was warmer in April. Okay, it was a longer heat wave compared to that. All right, I mean, this is the second one, but it's not 2 to 3 degrees as reported. You heard 2 to 3, some people say 2.5. It's compared to the average, it's about 1 degree warmer, sort of. And the other note that I made, and I come to the summary just at the end, is that the species is actually not very tolerant to temperature effects, according to scientific literature. So maybe the heat wave had no impact. So summary, I'm not sure what my time is, but should be all right. The blooms are common worldwide and recorded for over 100 years and way before the effects of global warming became dramatic in the last 10, 20 years. Long bloom durations are also common, unfortunately, longer than 2 months. Most likely fueled by nutrient-rich waters of the Bonny Coast upper lane, because it's away from any discharge of nutrients or from any big city influences. Slow spread into Gulf St. Vincent, and I put a little positive twist on that, because a fast spread would be absolutely devastating. Luckily, it has not impacted the Spencer Gulf, where the sardine hotspots are, for the tuna fisheries. And despite the fact that we had another marine heat wave in 2025, there's no obvious link between this extreme event and the toxic algae bloom. And that's when I want to finish. Thank you very much.
Thanks so much, Jochen. That's extremely interesting. You're getting a lot of little applause emojis there, so obviously everyone else is very interested, and I'm sure there'll be a lot of questions. For me, it was really interesting to learn that we actually rely on the algae blooms and how important that upwelling system is for our unique marine biodiversity in South Australia. And yeah, I'm hoping that we can touch on some questions about climate change and the storm on Monday and how that may have affected the mixing and the connectivity of the two gulfs. But yeah, we'll move along, and I just wanted to remind everyone again that if you've just joined, there's no chat tonight. We're using the Q&A, so hopefully you can see on your Teams there's a chat box, and then next to that there's a little Q&A. So if you've got any questions, please put them in there, and we'll try to get to as many as possible at the end of the session. But now we're going to welcome Brad Martin. Brad completed his honours and PhD at Flinders University, researching local shellfish reefs as fish habitats. Brad is now working as a project manager for OzFish, supporting fish habitat projects throughout South Australia and focusing on the restoration of seagrass beds and native oyster reefs. Brad has previously worked for the National Parks and Wildlife Service and continues to build strong community engagement to support aquatic habitat and restoration efforts. Brad will introduce to us tonight the Marine Mortalities Citizen Science effort and to let us know how we can get involved with that. So thanks so much, Brad, for speaking to us tonight.
Thanks, Alfie. I assume you can see the presentation?
Yeah, looks great.
Fantastic. So I guess I'm going to be talking about the algal bloom as a fish kill event, and from that perspective. Now, if you haven't heard of OzFish, we are a national not-for-profit and fishing conservation charity. So I'll talk in a bit later about, I guess, some of the solutions to trying to address some of the issues and impacts, and trying to sort of stay off some of the future algal blooms that we might see. So this year alone, we've already seen several fish kill events around the country. So a fish kill event is when we have unusually large numbers of fish or other aquatic organisms that die. And often that's associated with environmental stresses or these extreme events. So in South Australia, we're all quite familiar with the harmful algal bloom, but this can also happen with other natural disasters like bushfires, floods, and diseases. And these are typically really short events where people will just find a whole bunch of dead fish and then they disappear. And with climate change, it's forecasted that we might see more of these into the future. Now, waterway users like wreck fishers, divers, beach walkers, surfers are often the first ones to find these, fish kill events. So it's typically not governmental research, it's the public. It's probably the people who are tuning into this event who are quite familiar with some of this. And because these events can have really significant impacts to fish populations, it's really important to document them. And that information is relevant to governments, it's relevant to industries, and it's relevant to local communities. So we know that since at least mid-March, we've seen these massive die-offs of fish populations of various different species. We know that different species are impacted to different extents. So some species are getting impacted more than others for a variety of reasons. The algal bloom in many ways is kind of like an underwater bushfire. So it's suffocating fish, it's taking the oxygen out of the water, and it's producing toxins. And I know that there's going to be a fair bit of discussion about that a bit later. But these sort of environmental stresses can manifest as changes in fish populations. So they will try to move out of the way. You see different changes in their distribution, so they might not be around because of, say, a stressor in the environment, and also the lethal impacts on their survival. So recreational fishers and citizen scientists have been documenting this fish kill, I guess, really since it was first detected. Some of that's anecdotal, and some of that is been taking photos. And I think many of you are probably aware on social media, you've seen photos of all these dead and impacted fish. And that's really good. It's great to have that awareness at the community level. But I guess researchers sort of struggle to translate that information from social media into something that we can use to kind of extract data and numbers behind the impacts, turning what might be lots or many dead fish into workable numbers. And that's where citizen science efforts are really important. So pretty early on in the algal bloom, we turned to iNaturalist, which is a free online public global database and citizen science app that can be used to document, biodiversity. And in our case, we used it to document the impacted species. So it is a bit of a tool that we used. It's not necessarily designed for this purpose, but it's been pretty good, and you'll see shortly why. So if you're not familiar with iNaturalist, it's able to document photos of impacted species. So that's taking photos of any impacted species or just biodiversity, any living organisms you come across, uploading them onto the platform. So that's one record per species. And that provides information around time, date, and location. For any wreck fishers in the room, you can also obscure the location, so you don't have to give away your fishing spots. And it also automatically does that for any threatened species. It's not very good at documenting abundance. It's a much better tool at understanding the biodiversity or richness of species. So there's also a description option to provide that additional information so we can tease out those numbers. Those photos are publicly accessible to other users and essentially peer reviewed. So if you've got identification skills, if you know how to ID fish down to species, you can hop on there and you can help with identifying them. And those records then move across into all of these databases. So Atlas of Living Australia, which is basically run by the museums across Australia, as well as Global Biodiversity Information Database. And all that information is publicly available. So governments, researchers, and communities can all access it. And it means there's a permanent record. So early on in March, we set up an iNaturalist project, which is the SA Marine Mortality events for 2025. We've never really ran projects like this. So it's been a lot of learning on the go. And this project has been supported by researchers and volunteers. It's really community led in terms of both the uploads as well as the identification as well. So what this project does is it automatically gathers any records of marine animals within South Australia that are denoted as dead. And that means we can split that off from every live organisms. So all those records are occurrences. So like I said, we don't have abundance, but we have an idea of how many different species are impacted, where those impacts are, and this sort of project collates all of that information. And you'll see that on the slides here is some examples of what the project actually looks like, which is you can see all these numbers in terms of how many uploads are there, all of the number of species, etc. I do also need to highlight that death is still normal beyond the algal bloom, but we've got a pretty high confidence that the majority of the records are reflective of the impacts of the algal bloom. And that's because we know where the algal bloom roughly is. And we also know because of the sheer number of uploads that are coming through, some of the descriptions, and also like what you see here, people are coming across lots of different impacted species on their local beaches. And what that means is we're able to get all of that information and use that as data. And this is a bit of an example of what you can see over time. So what you can see on the graph here, this is mid-March. So many of you probably remember that's when those first reports from down at Waipinga started to come in. And since then, we've had this increase in the number of records over time. So this is records per day, and this is broken down by some of the different, I guess, groups of impacted species. And I'll get a bit into that a bit later. The other thing to note is this project is statewide. So you can see on the map that we do have records from other parts of the state. That was deliberate so that we can keep track of any other unusual deaths across the states. But as you can see, majority of the records occur in these impacted areas. And I'll talk a bit later about, I guess, some of the summaries that some of the information that we can pull out from this data set, and I guess some of the ecological solutions as well. So I'll talk a bit about sort of the positive next steps for everyone. Back over to you, Alfie.
Thanks so much, Brad. I love that analogy of an underwater bushfire. It really describes perfectly what's going on in our oceans at the moment. And it's really clear from what you said how important it is for community to be documenting when they notice unusual marine deaths, because we wouldn't know when these start otherwise. And I really hope that after tonight we can encourage a lot more of the people that have tuned in to participate in the recording effort. So next up, we're going to hear from Emeritus Professor Ian Gibbins. Until he retired in 2014, Ian was a neuroscientist and professor of anatomy at Flinders University, internationally recognised for his work on the microscopic organisation of nerves interacting with the skin and internal organs. He taught a wide range of topics across many disciplines and was heavily involved in bringing science to the public. He is now an award-winning video artist and poet, maintaining a strong interest in science and the environment, which informs much of his creative work. He has a lifelong love of the ocean, which he visits whenever conditions are suitable, which typically means during stormy conditions suitable for windsurfing. Ian will speak to us about the physiological impacts of Karenia species, including their neurotoxic and other systemic effects. Thanks, Ian.
Good. Thank you, Alfie, and hello, everybody. I'll try and get my screen sharing to work, and we'll go into this mode here, and we're going to show that one, and we're going to play, and hopefully you can see it.
Yeah. Oh, no, we can't, sorry. It just went back to you.
I'll try again. I just did exactly what I did last time.
Yeah, it was there until you pressed play, and then it disappeared.
Well, here we go.
Oh, hang on, we can see the Google Doc now.
The Google Doc. Hang on, I'll go back. Should be this one. You can't see this. I'll stop proceeding.
Give it another shot. Otherwise, I can get it up on my screen. Yep. So we can see that there.
So you're seeing now. If I go play, you should see the full screen.
Yes. Perfect.
Good.
I'll leave it to you.
Okay.
Thanks.
I'll just start my timer. So I'm going to tell you a bit about microalgal toxins and how they work. Before this event and before people started talking about it, I must say I knew nothing at all about microalgal toxins, but when some of the reports were coming out, I recognised that some of the toxins at least had actions on nerve cells, on parts of the nervous system that I understood well. So over the last little while, it's been a really interesting task finding out more about the microalgae themselves and the toxins that they produce, how they interact with various components of animals' body systems. So just to set things up in terms of summary, I'm only going to talk about the things which are best known, which in some cases is not very well at all. And there'll be other effects and there'll be other things that people might have noticed and which might have come up during other talks, which maybe we can come back to later. So the main effects that have been observed by the citizen scientists and by the formal surveys is for bony fish, that's teleos fish for the scientists. The most obvious things that they're dead. And there's very relatively few other physical effects that you'd notice on the fish, at least to the untrained eye. Sharks and rays, collectively they're called elasmobranchs. A lot of them have been turning up dead. And a surprising thing, as Janine mentioned, is that many of them have showing abnormally red skin, as though they've been blushing or something like that. Very unusual appearance. And then it's also been reported anecdotally that many of the sharks in particular have been showing abnormal swimming behaviour, especially coming in close inshore and not showing the normal sort of predatory or searching behaviour that we're more used to. Humans have reported a whole range of potential symptoms, the most common ones being eye irritation, a dry cough, and for some people nausea and diarrhoea. And I must admit I experienced that myself after windsurfing in Encounter Bay very early on in the days of the bloom. Another thing which has attracted people's attention has been birds washing up dead without any obvious physical effects on them. Birds are a bit harder to talk about because dead birds wash up a lot of the time anyway, and I don't know the data on whether there's more birds being washed up now compared to normal autumn storms, but that's maybe again something people can consider. So what could be causing all of this incredible range of effects on such a wide range of animals? Well, based on the species which have been identified so far, here's our most likely suspect list. So we're looking at toxins from two different species of Carenia, Carenia papilionacea and Carenia micomotoi, and probably several species of Carlodinium. I'll start with the ones which are in a way easiest to explain. So from Carenia papilionacea, they produce brevetoxins, and brevetoxins have been well studied, they're well understood now for many years. These are neurotoxins, and they work on one of the critical components of nerves and muscles, which gives them their activity, the electrical impulses which send messages down along nerves and the corresponding electrical impulses that are required for muscles to contract. Now, in general, neurotoxins can work in two different ways on the underlying biochemistry of the electrical generation by nerves and muscles. They can either block those processes or they can enhance them. Either way, it's a disaster for the animal. So brevetoxins work by actually increasing the activity of the nerves and the muscles. That can only be maintained for a short length of time. The nerves fire more than usual with different sorts of patterns compared to usual, and the muscles will contract more than usual, then everything seizes up. And that results in spasms in the muscles, paralysis, which can't be overcome by any sort of neural activity. And in some conditions, that will also result in numbness because the nerves are no longer transmitting any sort of information back to the brain. In a way, that numbness would be like having a local anaesthetic, although local anaesthetics work in a different way to the brevetoxins. The other potential effect of the brevetoxins is affecting a group of cells called monocytes. Now, monocytes are part of the white blood cell group, which are circulate in your blood, and which are involved in generating immune responses and especially inflammatory responses. So how your blood and your body responds to infection or a minor injury or something like that. And it seems that a different action of the brevetoxins, which is not related to their neurotoxic effect, is responsible for the respiratory irritation that so many people have experienced. The other toxins we've got on the list here, carlotoxins and chametoxins, are from a different group of the microalgae, the Calodinium species. And these are very, very strange compounds, and they have a very strange effect on the cells. So by way of explanation, all cells in your body are surrounded by a little membrane, a little sac, which keeps the contents of the cell in and keeps the rest of the world out. And a major component of that membrane are fats or lipids. And one of the most important ones is cholesterol. It's exactly the same fat that you don't want to have too much of in your blood, but it's absolutely essential for keeping your cells alive. And it turns out that the toxins from the Calodinium species are called carlotoxins, and there's chametoxins that work a slightly different way. Both of these toxins somehow interact with the cholesterol in the surface of the cells. What's that do? Well, it makes the cell membrane fall apart. And once that happens, or the components of the cells are no longer separated from the outside world, they all mix and the cell will die. And once the cell dies, and the tissue that the cells are made of will die, and depending what that tissue is, then the animal will die as well. The main Carenia species, which seems to have been reported in the bloom here in South Australia, current bloom, Carenia micomotii, has a dramatic effect on cells, very similar to that I've just described for the carlotoxins. It makes the cells fall apart, and that would lead to the death of the cells and the death of the surrounding tissues. But the component of the Carenia micomotii which does that is not well known. It's probably a fatty acid, but it requires the Carenia cells to actually be in contact with the cells which it's damaging. It requires direct contact between the cells and the tissues that are damaged. There's nothing being released into the water from those cells as far as we know. So what are the consequences of these toxins for fish, sharks and rays? Well, the brevetoxins will cause muscle spasms and paralysis due to the sustained electrical activity in the muscles and nerves. What does that mean? Well, the fish can't swim, can't pump water over their gills, so they'll suffocate, if nothing apart from anything else. The carlotoxins and the chametoxins and the Calodinium species destroy cells on the surface of the gills, which are the most sensitive surfaces of the fish which are exposed to the water. And so the gills are mostly responsible for the fish breathing, extracting oxygen and giving carbon dioxide back into the water. But they also have cells which make sure that the salt balance of the blood is maintained appropriately. So the salt concentration in the blood is different from the salt concentration in the surrounding ocean. And there are cells in the gills which maintain that balance. So if the gills are no longer functional, and they may well bleed out as a result of the destruction of the cells, then the animal is going to suffocate quickly. It may even bleed out through its gills. And also the salt balance in the blood will be disrupted, and that will affect all bodily functions in a very, very short time. For the Carenia micromotii, the Carenia cells, the microalgae themselves, get stuck in the gills and can clog up the gills just by their being there. But they also induce a lot of mucus secretion from the gills, and that clogs them up even more. And just like the calotoxins, the cells of the gills are destroyed and the gills are no longer work and may well bleed out as well. For sharks and rays, you have all of those things we've just described from the brevetoxins, the calotoxins and the Carenia micromotii effects. But there's possibly other things as well. So the neurotoxic effect of the brevetoxins potentially affects the electroreception, magnetic field reception of the sharks. So sharks can detect prey by the electrical activity of muscles in their target species, and they also can orient with regard to the magnetic field of the Earth. If the nerves going to those receptors are right near the surface of the skin, and they could easily be affected by brevetoxins. There's no evidence that no one's looked one way or the other. But if they were affected, that would lead to the sharks being highly disoriented with abnormal swimming behaviour. They wouldn't know where they were, and they wouldn't know where the prey, and they may be getting misleading signals about where prey is. And just like in the fish, you get destruction of the gills and potentially bleeding going beyond the gills into the circulation, especially in the small vessels of the skin and fins, resulting in blushing. But that's a guess. It's hard to know exactly what's happening there. Consequences for humans, well, the brevetoxins, the neurotoxins, can have sustained electrical activity in the nerves, leading to the nerves failing, and that would cause numbness in the exposed areas such as lips. May also contribute to effects in the airways and the gut, so bronchoconstriction, making it harder to breathe, and maybe some sort of nausea or diarrhea from the gut. There's good evidence that the activation of the monocytes, the white blood cells, leads to asthma and allergy-like effects in the airways, and they're partly due to the release of histamines. So antihistamines, just your common across-the-counter antihistamines, should work to reduce those effects. The calotoxins, the ones which destroy the cells, there's no direct evidence of this, but it'd be really surprising if they don't contribute to the irritant actions in the on eyes, nose, and throat. The calotoxins may spread via aerosol and get into your eyes and nose that way, but for the micromotii, you actually need to have your eyes in contact with the cells in the water itself to have any consequence of that, which may well be the case for surfers and swimmers. Now, a couple of other quick things just to finish off with. Indirect consequences of the toxins. The brevetoxins, the neurotoxins, are very, very stable compounds with a very long lifetime. They persist in the environment. If animals aren't killed by them but still absorb it, they remain in the liver of the animals for a long time. And many molluscs, including some edible ones, can store high levels of these toxins without being affected. Then eating the animals with high levels of stored toxin may cause paralysis and gut dysfunction in people, and even sometimes death, though that's rare. That's called neurotoxic shellfish poisoning. There's no long-term effects known for the calotoxins, probably because the high levels of exposure are so fatal that the fish don't get a chance to store them. But while people were talking earlier, there's some mysteries there which I haven't been able to find any answer to at all. So why are so many mollusks, the cephalopods, that's squid and octopuses, cuttlefish, pippies, scallops, why are they being affected? Lots of mollusks, different types of clams and oysters and things like that, can apparently absorb these toxins without being affected for it. But in this case, lots of them are being affected. Maybe it's damage to the gills of these animals by calotoxins. Maybe it's the neurotoxicity of the brevetoxins is just if there's high enough levels, the nerve cells of these animals would definitely be sensitive to the actions of the brevetoxins. But it could be either or both of those things or something else. There's all the other invertebrates too, which people have identified, the sea stars and sea cucumbers. I was surprised to hear that some of the corals are being affected as well. It's hard. neurotoxicity is unlikely to be a factor there, but direct contact with these toxins which cause cell damage could easily be something working there, and compounded by, of course, low oxygen levels in the water. And then finishing off with the mammals, with reports of seals with a flushing, similar to what's seen in the stingrays and sharks. It's very, very hard to understand that. It's almost certainly due to blood leaking out of the blood vessels and causing that effect. And the only way that can happen is by something which breaks down the walls of the blood vessels. Presumably one of these cytolytic toxins would do that. But how it gets into the blood without the animal dying in the first place is a very difficult thing to understand. So my guess is there's many, many things going on here, and trying to unravel them all will take a lot of time and a lot of careful research. So that's it from me.
Great. Thanks so much, Ian.
I'll
See a lot of little clap emojis there. Sorry, my dog's making noise. The effects of those toxins sound extremely unpleasant, and I hope that at least the numbing effect that you mentioned was present for all the animals that have died so far. And it sounds like we'll have to wait and see what effects this might have on local seafood industries with the potential for bioaccumulation. Yeah, maybe we'll hear more about that in the question time. And I just wanted to remind everyone again that if you've only joined recently, that the chat's not being monitored. Please put questions into the Q&A that you can find along the top bar of your Teams app or on your the web page you're on. Cool. So now we'll hear from Chloe Roberts. Chloe is a current PhD student at Flinders University, and tonight Chloe is going to discuss with us the movement of Port Jackson sharks and southern eagle rays in South Australia, and how the ecology of benthic sharks and rays could explain their high mortality rates during this algal bloom event. Thanks so much, Chloe.
Cool. All right, I'll share my screen. Okay, cool. Can we see that?
Yeah, looks good.
Right. Cool. Awesome. So yeah, I'm doing my PhD on some tracking of sharks and rays, particularly Port Jackson sharks and southern eagle rays here in South Australia. So I'm just going to talk a little bit about that and why we might be seeing some like really high mortality rates of those species. So chondrinthians is the group of animals that contains sharks, rays, skates, sawfish, and chimeras. They're a very diverse group of species. They arose a very long time ago, and they're generally slow-growing, long-lived species with low reproductive output and offspring survival. So they can be quite vulnerable to acute stresses like algal bloom or any other acute environmental condition. And 1/3 of these animals are already threatened with extinction due to mostly overfishing effects. So it's really important that we keep these animals in mind when we have these sort of big events, and think about how ways that we might be able to reduce the impacts on their populations. Here in South Australia, we have a lot of different shark and ray and chimera species. We've got the pelagic top predators that many are familiar with, particularly the white shark and hammerhead species. We've got coastal generalist species like the bronze whaler and the gummy shark. We have quite a few deep water species that a lot of people wouldn't be very familiar with, and they probably would not be seen by many people other than deep sea fisheries and stuff like that. But there's quite a few diverse species in that group. But probably the most abundant group of sharks and rays that we have in South Australia is our coastal benthic species. Things like the Port Jackson shark and the southern eagle ray, most of the other rays, wobbegongs and shovel nose and catfish, all sort of your coastal species that are foraging on the seafloor and sort of living close to the coast. So sharks and rays play really important roles in the ecosystem, and that's why it's really important to, keep an eye on their populations and do what we can to make sure that they're, their populations are healthy and not being impacted by things that we can control. So they play important roles in aiding decomposition and cleaning up the ocean through scavenging. They play really important roles in connecting ecosystems, particularly for those large species that move across multiple ocean basins. A lot of them modify their habitats, such as the eagle ray here in the video that's creating a feeding pit, that's recycling nutrients, and it's also creating microhabitats for other species. And then, of course, they play really direct roles in mediating prey populations and keeping prey under control. So I won't touch on the effects because Ian did such a great job touching on all of the different effects of the algal blooms, but I'll just jump straight into, what we've seen and why these animals might be affected. So we've seen a lot of Port Jackson sharks affected by this algal bloom and washing up. Interestingly, a lot of the records seem to be juveniles. So this one here looks like a juvenile. This one here looks very small as well. And so does this one in the bottom right. These look like very, very young individuals. And given, like I mentioned before, that sharks and rays have low reproductive output and offspring survival already, it's not surprising that the young and the juveniles could be most affected by this, given that they're, possibly the most vulnerable age class of the species. So if we look at some of the tracking data that I've got for some Port Jackson sharks here in South Australia, what we have found is that they have quite limited movements and they are quite restricted to very close to the coast. So these are two populations basically that I tagged at Christie's Beach and Long Spit, which is sort of a northern central area of the Gulf St. Vincent. And they actually had very limited movement. They stayed quite close to the coast, and even just between these two locations, which are only about 40 k's apart, there was no really overlap between the two populations. Individuals from Long Spit were not going to Christie's Beach and vice versa. And this is in stark contrast to Port Jackson sharks on the east coast, which have been found to travel up to 600 k's to another location and then returning back to where they were tagged. So they're making huge treks for such a relatively small coastal species. Then if we look at the southern eagle rays, which is another species that there's been a few reports of washing up. These all look like adults in these pictures, or near adults to me, but they're likely affected as well because they have even more restricted space use than the Port Jackson sharks. So I tagged individuals at two different locations, Seacliff and Christie's Beach, and you can see they've pretty much stayed at those locations for the whole monitoring period. So these are very limited space use for these animals. And again, this is different to a population of eagle rays that was tracked in Coffin Bay, which actually left the whole area of Coffin Bay in the cooler months and then returned. So they were making a lot more space use and taking bigger treks, probably not huge treks like the Port Jackson sharks on the east coast, but they were moving a lot more than our ones in the Gulf St. Vincent. We've also seen a lot of other elasmobranchs and sharks and rays affected in this bloom. In particular, the smooth ray over here, we've had a lot of reports of that one, the over 100 observations of these animals washing up. And then the southern fiddler ray as well, we've had over 300 observations of these animals washing up. And probably that number might be a lot bigger based on duplicate recordings and stuff like that. So these animals could be affected for a multitude of ways. These could also be animals that have restricted space use, like our Port Jackson shark and southern eagle rays. But another reason they could be affected is by their reliance on prey items that may be accumulating the toxins from the algae, but not dying from them. And then these animals, like the sharks and the rays, are eating these toxic prey items and then dying themselves. I do want to mention, however, that these are the animals that we are seeing. Those other animals that we might not see that are washing up that aren't washing up because they are deeper or they're further offshore, they could be affected, but we're just not quite familiar with that. So there is that possibility that other animals are affected, but it's likely that these animals potentially have restricted space use and are relying on prey that are heavily affected by the bloom. And that could be why that they are dying in such large numbers. So I just want to finish on some lighter, less negative information. So these animals are probably affected because they have restricted space use and they're eating these prey items that are accumulating things, or they're not moving away from areas affected by the bloom and they're succumbing to all of the impacts that Ian discussed. But many of the sharks and rays that are affected that we are seeing are listed as least concern, and they have stable populations here in Australia. So I'm hopeful that a lot of those populations may be able to withstand some of these impacts. We are continuing our monitoring of these animals. We are currently tracking and monitoring Port Jackson sharks, southern eagle rays, smooth rays, and fiddler rays. We just haven't got any results for the smooth rays and the fiddler rays yet. But we are continuing to monitor those species to see how they might be impacted. Really key things that we can take away, and I believe Brad and others are going to talk on this a bit more, is managing what we can in terms of what impacts the ocean. So managing our anthropogenic pollution, things like wastewater outflow and coastal development, that gives the animals and the ecosystems a chance to build up a tolerance and have a healthy environment to withstand these impacts. Things like restoring habitats like seagrass habitats that provide shelter and increase the resilience of ocean ecosystems. And of course, protecting our most threatened species so that when these events occur, they don't have the cumulative impact of other things like overfishing affecting their populations as well. So that's all of it for me. Thank you very much for listening, and thanks everyone for tuning in and being so interested to hear about what's going on and caring about our ocean.
Thanks so much, Chloe. Sharks are so often vilified by all of us and everyone's so afraid of them, but it's really lovely always to hear how important they are to maintain our marine ecosystems, and without them we wouldn't have the amazing underwater landscape that we have, especially in South Australia. And I really wanted to thank you for mentioning how important it is for us all to work toward reducing our own and other general anthropogenic impacts on our environment. And you often don't think about how what you're doing on land can impact the sea, but there's roles that we can all play, and I hope we hear more about that later tonight. So now we're going to pass back to Janine Baker, and she's going to tell us all about the potential impacts of the bloom on a range of particular species of concern and opportunities for the community to keep recording observations for citizen science. Thanks again, Janine.
Thank you. Okay. Thanks very much, Alfie. Just checking if people can hear me.
Yes.
Yep. Okay.
Yeah.
Thanks.
And you can see the slides.
Chloe and Brad have gone through some of the most common species that have been seen in the wash-up data so far. I want to mention the importance of not only recording what is found in your area, what you see on the beach or in the shallows, or even when you're out in the water fishing. It's very important that we get more information about what's happening on the seafloor, because there are quite a lot of things that would be impacted and are not washing up, and that would include things such as the heavier shells, attached fauna such as sponges, etc., unless they are dislodged by storms. There's a lot of benthic effect out there that we're starting to see now that the weather is changing and we're getting these storms, and lots of the seagrass and seaweeds are washing up. But those kind of impacts were happening during the summer when we didn't have that kind of weather. And I'll just start this section with some of the other uncommon species that are being seen in the data, in the iNaturalist data that people are recording. And this is quite unusual that we would see blue devilfish. This is one of the iconic long-lived, slow-growing, side-attached species here in South Australia. So work by Saunders in WA showed they can live for up to about 60 years, and they are very, very site attached. So they will stay in cave or under crevices for most of their life. They are very strongly pair bonded as well. The males and females live together, and because they are such a long-lived species, the breeding is intermittent. So they're not producing young every year. So for a long-lived, slow-growing species that guards the eggs in the cave, this kind of environmental impact can be very serious and have long-lived consequences for these. So we're only seeing a small number that are washed up, but in particular areas where previous research has an understanding of both the distribution and the relative abundance of these through previous dive surveys, it's important that those areas are resurveyed to determine the impact on populations. And there's quite a lot of Reef Life Survey transect data, for example, and also specific surveys for some of these long-lived, slow-growing species. So I would keep encouraging people to upload to the statewide marine mortalities database, and also the smaller ones. For Kangaroo Island, there are specific projects on iNaturalist as well for those. There was a question in the comments section which people will be chatting about later. One of them was the impacts on little penguins. Little penguins obviously are subject to multiple impacts and threats over time in South Australia. And overall, since the 1980s, populations have declined in a number of areas. There are many reasons for that. Including issues when they're out at sea. So there's feeding issues, there's attacks by predators when they're on land, and many other issues as well. But I just wanted to point out the fact that little penguins are washing up probably more often than what would be expected as part of the bloom. It's important that these carcasses are retained, because if there's any possibility of getting information about them, I think that would be helpful in the future. And Ian mentioned the possibility of bioaccumulation, and the research evidence certainly shows that seabirds and other air-breathing animals that are consuming algal contaminated fish can store the toxins. There's a number of research papers showing storage of toxins in not only the liver, but also kidneys, brain, heart, etc., and various tissues. So yeah, Granite Island population is down to about around 20 penguins. Same with Trowbridge Island. In the 1980s, there were thousands. I lived on Trowbridge Island for a year during the 1970s when there were thousands of these animals, and it's a real concern that populations have declined. So this is one area where research from the impacts of the algal bloom, I would say, is very much needed. Even though globally this is not a threatened species, individual populations certainly are. I wanted to mention lobster, and Brad will probably come back to some of these commercial species as well, because it's quite unusual what the data is showing. We did not actually expect that hundreds of dead lobsters would wash up along the foot of Yorke Peninsula, and that seems to be the case. So far there are only 26 documented on iNaturalist, but as Chloe mentioned, the data are opportunistic, so it just depends on what people are seeing at the time when they're walking along. There are anecdotal records of hundreds of dead lobsters along the coast. And there are some examples here of those washing up. So there are very many species, both commercial and non-commercial, that are washing up. it would be helpful to get carcasses of quite a number of different species over time. And I know that some people have provided these to government over time, particularly for marine mammals, have gone to national parks, and the sharks have been collected by Persa Fish Watch and transferred to SARDI. I can see Alfie there, which means it's time to finish.
Yeah, sorry, Janine, we're running out of time.
Yeah, sure. All right, thank you. So I just wanted to mention very quickly some uncommon species as well, such as black stingray. You can see the map here. So this is decades of records, hardly any black stingrays recorded here in South Australia, but some of them are washing up, not very many, certainly compared to things such as the more common smooth ray, where there's more than 100 records. This is the last slide, and I wanted to mention sea dragons because these are particularly sensitive animals. They were washing up on southern Yorke Peninsula even before the major wash-ups in April and May. So some of the earliest animals to wash up were the sea dragons, and that was the same on the Encounter Bay Surf Coast as well. So there's potential significant loss for sea dragon populations, particularly since they are fairly long-lived species for more than 10 years. We know from long-term work here in South Australia that not all individuals breed each year, even though there would be opportunity to do so. And unfortunately, these mass mortalities in several areas of South Australia have occurred prior to the spring and summer breeding period. So none of those adults, and many of them are adults that are washing up, none of those will contribute to the next generation. At the moment, we're seeking government approvals to transfer some of the carcasses legally to UTS University in New South Wales to do some ageing work, because they can actually age these using their tiny little otoliths, their ear bones, and get a lot of other information out of this. And I think someone in Kangaroo Island, might have been Ro Anna, said every life is important, and that is how we're approaching this. If people can record what they are seeing in their area, and potentially even gather things that are legal to gather. And I've got an example here for the fiddler rays. So Joe Barry at Port Vincent is happy to collect some and storage for future analysis, potentially of the toxins, but also for necropsy. So people around South Australia have agreed to do this, and that is a very good thing to do. I just wanted to finish and say thank you. This is a combined effort from very, very many people around South Australia. So there's a list of people who have been really at the forefront of the community effort over time, and I've put around 100 of the many iNaturalist contributors there as well. So I would say keep eyes out and upload as much information as you can. And we're also working with government and other volunteer organisations to try and get some subtitle monitoring done, because we've lost about 10 weeks of that already. And that, I just wanted to say thank you for that. I'll stop sharing now. Appreciate everything.
Thanks so much, Janine, and thanks for all the effort that you're pouring into this and this project over time. And you're right that every life does matter. So on that note, we're going to pass back to Brad, and he's going to share a summary of the iNaturalist marine mortalities project data and the importance of our ongoing monitoring and what we can do as community members to aid the recovery efforts. Thanks, Brad.
Cool. You see the slides?
Yeah, all good.
Fantastic. So as you've just heard, this information that's already been gathered is being really valuable for understanding individual species. And I'm going to zoom out to provide a bit of an understanding of what we know from the ecosystem and from the fish habitats. So since mid-March, which is when majority of, I think, the public has become aware of the algal blooms, there's over 3,000 uploads. They represent a dead organism or species in a given location and time. So we know the actual numbers is far, far greater, likely to be in the tens of thousands or millions if we're looking at across different species. We know that majority of the uploads, most of those records are fish. So that's ray fin fish, and this chunk of 25% is sharks and rays. So that's what's most commonly being recorded. This stat of over 200 species that are impacted, this has come from the iNaturalist data. Not sure whether slides are playing up, but more or less similar situation. About half of the recorded species impacted are fish, and lots of different types of fish as well. There's also a big chunk of other, which is almost about 40% of the impacted species. And a lot of that is marine invertebrates. So that's all the diversity of snails, worms, crustaceans, and all of these other parts of the ecosystem. And that's why it's really important to document all of these impacted species, because it provides beneficial information. This here is the most commonly recorded species. And as you've heard, a big chunk of that are these benthic rays and sharks, reef fishes, crabs, and really iconic species. What we know from a functional point of view is that they do share common traits. So most of the species that seem to be impacted are seafloor species. They're hanging around on the seafloor, or they're benthic associated, and they typically aren't very mobile. So they mainly have small home ranges, or their natural defense is to hide. So this means they're really susceptible and vulnerable to the algal bloom. And we also know that from what's not appearing in the citizen science data. We also know from the diversity of species that this algal bloom is impacting fish communities in multiple habitat types, particularly macroalgae reefs, which are our kelp forests or the Great Southern Reef. We know that amongst the impacted species are seagrass meadows, and we know that from weedy whiting, dusky moorwong, other seagrass species, and similar with bare sand flats. There's a lot of species that hang out under sand that are washing up in large numbers. We also unfortunately know that the algal bloom is impacting multiple age groups of fish. So everything from small fry and fingerlings through to large adults. So there's potential for long-term population impacts. One of the other things we know by documenting all of these different species is a big chunk of what's washing up are prey. So that's all the worms and shellfish and gastropods and small fish. So that's going to have implications for the food web in these impacted species. Areas. As it's also been mentioned, we also know that some recreational and commercial species are impacted, particularly those that, again, are associated with the seafloor, benthic species like lobsters, crabs, and flatheads, and things like shellfish, which aren't very mobile. One of the interesting things we also know from the dataset is there's very few records of our common recreational species, so things like salmon, whiting, and snapper, which hopefully is a good sign that they're actually being mobile and moving away from the algal bloom. So we know that the algal bloom isn't necessarily having significant impacts across all of our marine life. It's not a complete wipeout, but we also know that there's a lot of information that we just don't have in terms of whether there's hidden impacts offshore to these populations or to the habitats themselves, as sort of been brought up a fair bit in these other presentations. Make some sort of general estimates around recovery based on different life history. So we also know that some fish will recover quite quickly. Species that are really mobile, that produce a lot of offspring, and that are habitat generalists, these are likely to move back into impacted areas quite quickly. We also know that some species might take years for their populations to bounce back, and that's because they have small home ranges, not very mobile, produce offspring. So going forward, we're going to see this uneven recovery in marine areas that are impacted by the bloom. Now, importantly, there are a range of options to help this situation. So first and foremost, starting with monitoring itself. So if you've got photos from the past couple of months, please feel free to upload them onto iNaturalist. And that includes some continuing monitoring in any impacted areas where it's safe to do so. As mentioned, there's a lot of unknowns, particularly what's happening underneath the water. Most of the records have come from what's washing up on shore, where it's nice and accessible. If you're coming across any unusual fish kills, report them to Fish Watch. Get in contact with PIRSA, let them know, and that's going to provide the best opportunity to figure out any community responses and to collect any specimens. I also really want to highlight that there are historic and ongoing stresses in the marine environments. The algal bloom is another stressor. And when you get enough of these stresses, it causes a decrease in survivability in fish. And that's why we're seeing these fish kill events. So a way to address that, particularly for the next, trying to stop the next algal bloom, is restoring, maintaining and conserving our fish habitats and our marine ecosystems. And that's both in the coastal environments, which is impacted, but also in our freshwater waterways. Addressing pollutants and stresses that are coming down the system and potentially impacting the coast. So recognizing that the coastal environment is connected, which I think quite a few of our speakers have talked on. There's also the opportunity to donate and get involved. So whether that's volunteering your time, providing financial contributions to NGOs and organizations active in this space, as well as resources as well. And there's a range of advocacy organizations who are involved with communicating the impacts and communicating some of those solutions. So I recommend getting involved. I've just included a couple, but we know there are heaps of organizations that are involved. So just get in touch, reach out, and lend your fish a hand. I also really want to talk about this long-term ecological solutions to addressing stresses in the environment and fish kills. So we know that with climate change that we're expecting to see more of these negative stresses in the environment as we continue to move away from the normal. And there are ways to address that, and that is maintaining and restoring our aquatic ecosystems. So habitats like seagrass meadows, like shellfish reefs, and like our freshwater riparian vegetation and creeks. If we have healthy fish habitats, we get more fish, and we also have these positive feedback loops. We have these inbuilt ecological solutions to try and reduce these stresses. It also provides more food, habitat, and shelter. So when these events happen, our fish populations can bounce back quicker and faster and have more available options to survive these events. So I'll leave up all of these solutions, and I also do really want to thank all of the OzFish volunteers and all of the citizen scientists who have been supporting the iNaturalist data and who are continuing to support our projects going into the future. And if you want to learn more about OzFish, please look us up. Please come along and get involved. As mentioned, we do a lot of seagrass and shellfish work, and we'll certainly be getting back into the water to do that work later this year.
Thanks so much, Brad. That was great. Yeah, and please leave that up, that the opportunities for people to get involved, because I think it's really important, and it is really impressive the restoration work that you do and the OzFish does throughout Australia. And I think it's an amazing and empowering way for people to connect with marine ecosystems locally. So I highly encourage everyone to get in touch with Brad and OzFish. So now we're going to move on to questions. I'm sure there's going to be a lot, and we've had a lot come through already. So we'll do our best to get through some and try and stick to ones, like especially try and get through ones that have been asked a few times. So Brad, while you're fresh in speaking mode, I might ask one of you. There's been a lot of questions around, are there dangers to animals and humans if they're eating the fish and marine life that have been affected by the algal bloom?
Yeah, so there's definitely health advice. Basically, if you're coming across dead shellfish or dead fish, don't eat it. It's not going to be great bait. And there is that concern around, I mean, any dead or dying animals, you can get sick from not just sort of any algal blooms, but other bacteria and other microorganisms in the system. So long story short, don't use it for consumption or for bait. I also recommend supporting your fishing stores, supporting aquaculture industries as well. What you're going to get through your shops is going to be perfectly safe to use. The other thing I'd recommend is if you are concerned around the impacts, fishing areas that aren't impacted by the algal bloom, not just in terms of reducing the potential issues around consumption, but also to minimize your exposure. And thankfully, the Spencer Gulf certainly hasn't seen this large algal bloom.
Yeah. And did Ian or anyone else want to talk about that, or we can move on to more questions? Maybe if all the presenters pop their videos and mics back on. We're getting some questions now about the storm, how the storm has affected the algal bloom. Maybe Jochen, are you able to talk to that?
I mean, any mixing helps to disperse the algae and reduce the concentration. You know, but it's a little bit difficult to speculate into the future. You know, it's also getting colder now. I don't know. We have to wait and see.
Yeah. Yeah, and I suppose it's such a unusually fierce storm that we had, so it can't really be easily compared to the conditions that we're seeing in past years. We'll move along to some more questions. They're coming through. For Ian, there's some questions around, is the reddening of the sharks and rays due to the hemolysin that the scientific literature say it makes, the Mikimotoi?
Yeah, this is a very good question. It gets technical, so I'll try and make it as simple as possible. So what a hemolysin, what that means is it's something which breaks down the red blood cells. So as I said in my talk, all cells are surrounded by a membrane, and inside the cell, it's all the components of the cell which make it work. On the outside is the rest of the world. If it's a blood cell in the blood, that's the blood plasma. But the composition of the blood plasma is completely different from the composition of the material inside the cell. So when the cell lyses means it breaks, and all the stuff inside the cell can mix with all the stuff outside the cell. If it's a red blood cell, the stuff which makes the red blood cells red is hemoglobin, the chemical which transports the oxygen around the bloodstream, and which makes the blood look red. When the blood cells burst, the hemoglobin come out of the cells and go into the blood, and that's called hemolysis. Now, that doesn't happen very often. It's a very, very rare event, sort of pathologically, because in order for a chemical to get to the blood cells, it's got to get into the blood somehow, if you're in an intact animal. And it doesn't happen very often, though some diseases can cause hemolysis. So, but on the other hand, it's a very easy test to do in a laboratory. It's very easy to get some blood samples and mix them up with a chemical or some biological agent and see if the blood cells burst or not. And so to look at the effect of a toxin or some other agent on the integrity of the membrane that surrounds the blood cells. So lots of tests use hemolysis, the breaking of red blood cells, to see how chemicals in general and toxins in particular can interact with the surface of a cell. Now, when you look at that blushing in the sharks, it could be by hemolysis. It could be that something's got into the blood and burst all of the blood cells. But if that was the only thing which happened, the animals wouldn't look any different, because all that blood would still be, that hemoglobin would still be held within the blood vessels. So something else has to have happened. Somehow the blood has had to have got out of the blood vessels into the surrounding tissues, like when you have a bruise. So you get a bruise, you impact to your skin, all the little tiny blood vessels in your skin burst, the blood leaks out, and initially anyway it looks red and then eventually goes black as the blood clots and starts to break down. Now, two things about the red looking sharks is that they still look red. So that means it's presumably it's relatively fresh. The hemoglobin hasn't broken down. The red pigment in the cells hasn't broken down. But it also means it must be outside the blood vessels. And it's some careful microscopic pathology should be able to tell you that and look at the details of what's happened to the blood vessels. But my guess is that it's a secondary effect of one of these toxins not the neurotoxins, but the other sorts of toxins which break down cells. And my guess is it's gone in through the gills and into the circulation, and then the circulation's broken down, and it'd be, hopefully the animal's already mostly dead by then.
Yeah.
Because they'll lose blood pressure, they won't be able to supply oxygen to the blood vessels properly, and they would die effectively of lack of oxygen.
Yeah, that's very unpleasant.
Yes, yeah. But there might be some, it's a very unusual effect, and it requires some pretty careful pathology to look at that, I think. Yeah.
We might move on to another question. This one maybe for Chloe. So given that multiple housing developments are planned and already underway along the South Coast, Goolwa and Victor, how is the increased runoff and stormwater likely to exacerbate conditions for toxic algal blooms in the next few years?
Yeah, this definitely relates to what we've got on screen right now with Brad's great presentation. Coastal development and any sort of anthropogenic impact that we're placing on the oceans, whether it be stormwater outflow or degrading the coastline to create houses, is going to have some sort of effect, and then that can accumulate and compound with the effects of climate change and reduce the resilience of the environment to withstand acute stress like a algal bloom. So ways we can sort of address that is engaging in these restoration programs like Brad has mentioned, engaging in ongoing monitoring, and really putting the pressure on developments where possible to make sure that they're engaging in sustainable practices and monitoring where possible. So yeah, it's definitely possible that coastal development could have an impact, but there are protocols in place to hopefully minimize those impacts, and it's just about making sure that we all engage in restoration and research and advocate where possible to kind of reduce the stresses on the environment where we can. Yeah,
definitely. We've got another question here, maybe for Paul. So for divers who want to get involved and monitor coastlines, people are wondering whether it's safe to get in the water at the moment and do monitoring.
Thanks, that's a great question. I know the state government has put out an advice to say don't enter the water if it's obvious that the algal bloom is present, but how do you know whether it is or not? Going back to that first dive I did on the 6th of April, we actually called the dive. I started getting a sore throat after about half an hour into it. It was a really bad sore throat. So I motioned for my dive buddy and partner Elizabeth that we should get out, and we did. She had no symptoms. That lasted about a week. Whether it was all related or not, I can't confirm. But I did take antihistamines and it did clear up eventually. I then got back in the 18th, no symptoms. I'm comfortable getting back in, but as a dive guide and dive professional, I haven't been taking people into it when I've known it's there, just because of the risk factors. So I'm not sure I can really answer the question to the satisfaction that's required, but I think if it's obvious the algal bloom's around, or you can tell from the heat maps, etc., that it might be around, then it's probably best not to get in.
Yeah, there's evidence there on the beaches. Oh, sorry, Janine.
Yeah, I wanted to mention something about that as well, because, and thank you, Paul, I agree that people should be only in the water where it's safe to do so, but in areas where the bloom has passed, it would be good to see some coordinated efforts from both government and the university and the community as well, because there are a lot of trained community divers out there, for example, through Reef Life Survey, who some of them have already put their hand up and said we are very happy to go out and run transects in some of these areas. There's opportunity also for underwater drone work. I think that's quite important, particularly in less accessible and areas that are not as safe, and also remote underwater video, and also just even towed GoPro from boats. Those kind of things are very, very helpful, and they're going to give us a lot more indication of what's happening out there. And on that point of benthic impacts, I also wanted to support everything that other speakers, particularly Brad, has said tonight about restoration. It is absolutely essential, and it's something that we really have to look at seriously in the 21st century, in the 2020s and beyond. All major benthic habitats, and we've got a great program here in South Australia for seagrass restoration and also for oyster reef restoration. The next phase is going to be canopy seaweed restoration, and that's occurring in other parts of Southern Australia. It's very important because there are significant areas of macroalgal loss, not only through this, probably through this algal bloom, but also over the past probably 2 decades since we've done early survey work. So, all right, I'm off topic now. Thank you.
And on that note, Janine, I think as Chloe said, I think terrestrial restoration efforts are probably equally as important because the runoff does affect the marine
absolutely
ecosystems. Yeah. We are running out of time, but I'm trying to get through some more questions. So we've got a lot of questions about the neurological toxins and their effects on the sharks and how the sharks have been behaving strangely. Maybe if Ian or Chloe could speak to that. Oh, you're muted, Ian.
Do you want to go first, Chloe, on the behaviour? Because mine's just a theory, not a genuine explanation.
Okay, well, mine's kind of just a theory as well, but yeah, look, sharks and rays and even white sharks, they do occur along our coasts all year round. We have tracking data that proves that these animals are within close proximity to the coast. All throughout the year at various random times. There doesn't seem to be any patterns predicting when these animals come in. They could be performing strangely for a plethora of reasons. It could be the neurological effects. It could be that they're seeking waters that are potentially safer and either don't have the bloom, or they're searching for more oxygenated waters. It's really hard to tell what is causing these strange things that we've been seeing in our animals. But yeah, hopefully we'll, that will sort of start to subside, and we won't, with the change in weather, that we'll start to see changes, and won't have so many strange interactions. But I don't know what it could be exactly. I think there's a wide range of things. But yeah, perhaps Ian can elaborate on it a bit more.
Well, my idea was looking at some of the footage of the behaviour of the sharks in close, and the fact that so many sharks were coming into such shallow water, including all the places where we go surfing and stuff. And a really incredible feature of sharks and rays is that they can detect the electrical currents of the movement generated by the movements of fish around them. And they also can detect the magnetic field of the Earth, and so they can navigate towards prey fish. So especially rays can detect creatures living in the sand, but beneath the surface of the sand, by the movement of their muscles, by electrical currents generated by them. And then the longer distance migrations of the sharks in particular seems to be that they're able to read the Earth's geomagnetic field like a compass. Now, the way they do that are these amazing little structures on their along their face, called ampullae of Lorenzini, if you want to look it up. And I didn't think anything much about that, except they provide a direct route of things potentially of things in the water through to the receptors. So they have these little canals, and they're in. They exchange with the water, the surrounding water. If there were toxins in that water, if the toxins are soluble in the water which has access to the receptors, the neurotoxins the first thing they would hit after the receptor is the nerves going from the receptors to the brain. It'd be like having your eyes or your ears or your fingertips addled. You'd be getting sort of the wrong sorts of sensations coming. It'd be like seeing stars in your eyes or ringing in your ears or tingling in your fingertips or something like that coming from these receptors. I have no idea if that's true or not, but it's feasible in terms of the underlying chemistry and biochemistry of those receptors.
Yeah. We'll keep moving along. We've got a pressing question that I think is on a lot of people's lips. It's about the effects of the desalination plant at Lonsdale and Kangaroo Island and the impact that they might be having. I thought maybe Janine, I know you've got some slides. Maybe we could see.
Yes. Okay. So I'm just going to share screen now. Here we go. Okay. Can you see the screen now?
See the southern blue devil.
Yes, so yeah. I'm going to just go into this. So that is a very good question because, and I'm hoping that Jochen can talk to this as well, because over the past 3 months, we've got lots of different hypotheses coming through because people are making correlations with what's happening in other parts of the marine environment. And as we always say, correlation is not necessarily causation. And Jochen has shown that this is a frontal bloom that would have started in the southeast system in association with the upwelling, and it has moved in a northwesterly direction. The actual first impacts were in the lower southeast, so we're talking hundreds of miles from any desal plant and actually in the opposite direction of current flow as well. So there is no evidence that there were any deaths at the time in the desal plant area of the Lonsdale outfall, which the Lonsdale and also Kangaroo Island desal plants are monitored regularly. And all of the deaths of thousands and thousands of animals that were being seen around South Australia were nowhere near the desal plants. And also wanted to mention, Ian mentioned some of these, some of the conditions for these species as well. Jochen did. So Mikimotoi, the growth is inhibited at higher salinities. And both of these species, in particular, Mikimotoi, as Jochen mentioned, blooms in normal oceanic salinity, but also in lower salinity conditions, such as after flood events. So there's a number of areas where there have been both upwellings and also floods. So two completely different type of conditions, but they have both created very, very serious Mikimotoi blooms overseas. Then they had nothing to do with higher salinity conditions. So on that point, I also wanted to include this slide from Perry Coleman, who's an environmental consultant, has been studying Gulf St Vincent for decades and knows an enormous amount about the functioning of Gulf St Vincent. Perry provided some stats to help us answer this question. So in terms of the natural evaporative processes in Gulf St Vincent, from the salt marsh and mangrove areas and the salty brine that is exported from the Gulf every year, it's estimated to be about 270 million tonnes. So even at full capacity, Lonsdale is producing about 1% of that amount of salt export. So we're talking completely different orders of magnitude here in terms of the salt, the salt that is being provided back into the Gulf from the Lonsdale plant. And there's no evidence that there were any major fish kills in that area off the Lonsdale plant. Hopefully that is enough. Maybe Jochen could mention more. I'm not sure, but yes.
Jochen's cat might have something to say by the looks of things.
Jochen's cat.
It is cat.
He knows.
Okay, I can say just briefly a little bit. I mean, desalination discharges come with their own problems. They can cause harm on a relatively short spatial scale, say within a kilometer or so. And so, but they're in this case have absolutely nothing to do with the event. And if there was something happening near desalination plant, I will probably be one of the first people putting my hand up and say, "Hey, you know." But,
I agree.
But there's no apparent, no obvious link between the diesel discharge and this toxic algae bloom.
Yeah.
My cat ran away. I can't show my cat again.
I think we might leave it there tonight, just because we are running over time, and you guys have poured so much time into this already. So apologies to those. There were a lot of questions coming through, and apologies to all of those that we didn't get to. But hopefully, you know a bit more. You have a bit of a better understanding of the science of this algal bloom now. And if you do have any absolutely pressing, dying questions, please email them through to me, and I'll do my best to get them answered. But I'd like to say a big thank you to all of our presenters tonight. It's incredible the amount of time and effort you're putting into keeping the community informed about this. And it's really, really great to see. And I think that we really appreciate everything that you're doing. So thank you very much. And we'll leave it there.
Thank you, everybody.
Big claps for all of you.
Thank you. We're happy to answer questions as well, Alfie, in the future. To if people send them to you, you're welcome to send them on. Yeah. Okay.
Thank you, Alfie and the Centre, for arranging all this. It's been fantastic.
Yes, I agree. I wanted to say thank you to Port Environment Centre too, and Alfie. Fantastic moderation, very good, and Janice and the person who I can't see who's there as well. So thank you all very much for your time and effort, particularly out of hours as well. Yeah. Must be beer o'clock. Okay. Thank you. Goodbye.
Right. Bye.
Bye.
Bye. Thank you all.